Video is made by Mia Donnenberg and Samantha Chong, and this is video number 19. Today we will be going over the testosterone signaling pathway, including the specific properties of the pathway. Testosterone signals can be translated directly to changes in gene expression, which is the classical pathway, or testosterone can activate kinases that may regulate processes required to maintain spermatogenesis, which is a non-classical pathway. In this video, we will focus on the classical pathway.
The testosterone signaling pathway starts in the cytoplasm and moves into the nucleus. Testosterone diffuses through the plasma membrane and binds to the androgen receptor in the cytoplasm in the cell. The signaling molecule, testosterone, is hydrophobic because the intracellular androgen receptors are inside the cell, and it can pass through the phospholipid bilayer. The tails of the bilayer are hydrophobic while the heads are hydrophilic, so it permits the passage of the hydrophobic testosterone. Once the binding of the testosterone and the androgen receptor happens, there is a conformational change in the receptor that allows the receptor to be released due to heat shock proteins. Androgen receptors allow the body to respond appropriately to hormones. The receptors are present in many of the body's tissues where they attach to androgens. The resulting androgen receptor complex then binds to DNA in the nucleus and regulates the activity of androgen responsive genes.
Another purpose of the testosterone signaling pathway is the building of muscle, bone mass, and blood cell production. An advantage of androgen receptors are that they mediate the cellular actions of androgen and are helpful in maintaining the balance of hormones. A disadvantage of androgen receptors is that the amplification of the androgen receptor leads to an overexpression in the androgen receptor, which could lead to mutations and increase the chances of an individual getting prostate cancer. In the testosterone signaling pathway, testosterone, which is a ligand, binds with the androgen receptor and releases a response in the cell that is mostly related to DNA expression. After the binding of testosterone, the body responds by turning on genes to synthesize proteins, which are in charge of making the features that differentiate the anatomical features between male and female babies. The proteins produced help to navigate the formation of the ducts and passageway that will bring the sperm. This also begins the regulation of gene transcription. In response, the development of male sexual characteristics begins.
In order to terminate the testosterone response, the follicle-stimulating hormone is released. This hormone is produced and secreted by the anterior pituitary. The FSH signaling pathway occurs on the Sertoli cells of the testes and regulates the production of testosterone. The receptors of these hormones are called follicle-stimulating hormone receptors, or FSH receptors. These hormones can act as antagonists and agonists since they increase or decrease cellular activity as needed. Once the hypothalamus alerts the pituitary gland, it produces the follicle-stimulating hormones, which travel in the bloodstream to the gonads and stimulates the production slash release of testosterone.
An agonist of the testosterone signaling pathway is luteinizing hormone releasing hormone inverse agonists, which is abbreviated to LHRH. LHRH agonist is a hormone that is made by the hypothalamus and is endogenous because it is produced by the body. In the testosterone signaling pathway, the making of hormones are blocked by LHRH, and the agonist binds to the androgen receptor instead of testosterone. The cell response is changed because LHRH agonists reduce the amount of testosterone made by the testicles. LHRH drugs can be used as well, especially by men who have prostate cancer.
Thank you for listening about the testosterone signaling pathway.